Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
If you follow the world of viral research and immunology, you know that modern medicine has already achieved something. Well, something truly monumental in the fight against HIV.
Absolutely. The advent of combination antiretroviral therapy, or CART, just completely shifted the landscape.
It really did. It took HIV from what was essentially a rapid death sentence and turned it into a manageable chronic health condition.
And this therapy is so effective that it can suppress the virus to undetectable levels in the blood, allowing millions of people to live long, healthy lives.
But here's the thing. Here is the enduring challenge, the paradox that we're really diving into today.
That CART doesn't and really cannot eradicate the virus.
Exactly. The virus is still there. It remains hidden.
It nests away in what scientists call viral reservoirs.
And what are these reservoirs exactly?
They're dormant cells, mostly a specific subset of memory T cells, and they're scattered throughout the body, particularly in places like the lymphoid tissues.
So they're like these silent ticking clocks, just waiting for therapy to stop so they can reactivate.
And trigger a massive, often fatal viral rebound.
Yes. And there's a second mystery here, one that's just as frustrating. Even with the virus suppressed, this chronic inflammation and immune dysfunction, it just persists in people living with HIV.
It does. It's a low-grade, simmering inflammation that contributes to all sorts of long-term health issues.
Things like heart disease, neurological problems, bone density loss.
Right. What researchers often call accelerated aging.
So the big question is why? Why does the immune system stay exhausted, and why does this reservoir stay intact, protected in this sort of subtly toxic environment?
Well, that's exactly what we're digging into. We're looking at a new study published in Science Translational Medicine by researchers at the Institute of Human Virology.
At the University of Maryland School of Medicine, led by Dr. Guangming Li and Dr. Lishan Su.
And their mission was to get to the bottom of this, understand why both the inflammation and the reservoir persist, even with effective CART.
And what they uncovered is, it's really striking. They found that a crucial part of the immune system's own attempt to fight the virus is, in a way, self-sabotaging.
Actively working against the body's chance for a cure.
And it all comes down to a single, very rare, but absolutely critical immune cell.
A tiny warrior that essentially turns into a kind of double agent.
Driving the very chronic inflammation and immune suppression that HIV needs to hang around. We are talking about the plasmacytoid dendritic cell.
And that, right there, captures the core tension. We've had this incredible success with CART, but the term we often use is a functional cure.
Meaning it controls the virus, but it doesn't actually cure the disease.
Precisely. The persistence of that viral reservoir means we haven't achieved a sterilizing eradication. And, just as critically, we haven't achieved complete immune recovery. The body is healthier, but the immune system is still deeply damaged.
So if CART is keeping the viral load down in the blood, why are people living with HIV still dealing with this suboptimal immune system? Why are they struggling with these inflammatory diseases that aren't even AIDS defining?
Well, the persistence of those reservoir cells is directly, and I mean directly, linked to this failure of the immune system to fully recover. The body is just stuck in a state of chronic, low-level immune activation.
A perpetual red alert.
That's a perfect way to put it. It's long past the acute infection stage, but the alarm is still on. And the key mechanism the researchers honed in on is the sustained activity of the type I interferon signaling pathways.
Type I interferon, or IFNI. Remind us, you can think of that as the master alarm bell of the innate immune system, right?
Exactly, it's the master alarm. And the cell that is primarily responsible for ringing this alarm, particularly in the context of a chronic infection, is the plasmacytoid dendritic cell, the PDC.
So let's unpack that cell, the PDC itself, because this is where that double agent analogy really starts to make sense. In a healthy system, say fighting the flu, these PDCs are the heroes, aren't they?
Oh, absolutely. They are critical for our early defense. When your body sees an acute viral threat, the PDCs just swarm the site and produce massive amounts of this type I interferon.
And that's the first line of defense.
It is. That IFNI alerts all the neighboring cells to prepare for an attack. It activates other immune cells, and it mobilizes the entire adaptive immune response. It's the emergency broadcast system, and it's essential for getting a virus under control quickly.
But in the context of chronic HIV infection, this essential function somehow turns against the host. How does HIV manage to trick or, overstimulate these cells?
This is the absolute crux of the pathology. In chronic HIV, even under suppressive CART, these PDCs become persistently overactivated.
And the study found they become enriched. They cluster in the lymphoid tissues, right?
Exactly, in the lymph nodes and the spleen, which is precisely where the viral reservoir primarily hides.
And HIV doesn't even need to be replicating wildly to make this happen.
Not at all. Residual HIV particles, or even just viral components from dying infected cells, can be taken up by the PDCs. Once that viral material is inside, an internal sensor called toll-like receptor 7, or TLR7, recognizes it.
And PDCs have a lot of this TLR7 sensor.
A very high expression of it, and that detection mechanism is what triggers the continuous chronic low-level production of type I interferon.
So the PDCs are constantly shouting, "Danger! Danger!" But instead of helping, this chronic noise is what's driving the harmful immune inflammation.
Exactly right. And we know that while CART does reduce the IFNI response compared to an untreated infection, those levels remain elevated in people living with HIV compared to people who don't have the virus. And that creates this toxic inflammatory environment.
And what does that constant toxic environment do to the specialized T cells, the actual soldiers that are supposed to be finding and eliminating the reservoir?
Well, the constant IFNI signaling acts as a chronic stressor. It's like a biological white noise that drives this inappropriate T cell differentiation and hyperactivation.
And you can measure this, right, with specific markers.
We can. We look for markers like CD38 and HLA-DR being co-expressed on the cells. This tells us they're in a state of continuous non-productive proliferation and inflammation. The T cells are constantly being told to activate, to divide, but they can't clear the infection. They just end up hitting a wall.
They become exhausted.
Precisely. This constant activation pushes the virus-fighting T cells toward a state of T cell exhaustion. They start expressing inhibitory receptors like PD-1 and TIM-3. They lose their ability to produce key antiviral molecules, and they undergo increased rates of what's called apoptosis.
The programmed cell death.
Exactly. They just run out of steam. They become deafened by the constant alarm, and this self-sabotaging loop continues, which in turn protects the viral reservoir.
That sets the perfect stage for the research then. The scientists at the IHV needed to interrupt this specific cycle. The hypothesis was clear. If they could silence the alarm, the PDCs, then the fighters, the T cells, would recover and attack the reservoir. So what was the game plan? How did they test this theory?
They focused on interrupting this PDC cycle directly, but in a living system that accurately mimics the human pathology. So they used these humanized mouse models.
Specifically the HuBLT mice.
Oh.
Bone marrow, liver, thymus.
Right. And these models are crucial because, just like in people living with HIV, CART works very efficiently to inhibit the virus, but the reservoir still persists and that inflammation is only partially suppressed.
And their intervention was very targeted.
Highly targeted. The goal was to reduce or deplete the overactive PDCs using a specific tool, an antibody known as anti-BDCA2, which the paper calls 15B. The BDCA2 receptor is expressed exclusively on human PDCs, so it's a perfect target for a surgical strike.
Now, in some experiments, they used a toxin conjugate, this 15B SAP. Why do that? Why not just use the antibody to block the function?
That's a great methodological question. Well, blocking the function with just the antibody can inhibit the signaling. Using the toxin conjugate, which links the 15B antibody to a seborrheic toxin, allows for the rapid and highly efficient elimination of the PDCs.
So for a proof of concept study, they wanted to be absolutely sure. Remove the cell entirely and see if that resolves the problem.
Exactly. The goal was to clear the double agents completely out of the system.
Okay, so they're silencing the alarm bell to wake up the main fighters. And the study focused on a very specific subset of T cells, which they call the elite fighters. We should probably spend a moment here. Can you define these stem-like CD8 plus T cells and explain why they're so critical?
Absolutely. The study specifically focused on what they term stem-like CD8 plus T cells, or CD8 plus TSL cells. And these cells are critical because they have this sort of progenitor-like activity. They can self-renew over the long term, and they're essential for controlling chronic viral infections and even for fighting cancer.
And they're defined by a very specific set of markers on their surface.
They are. They're positive for a transcription factor called TCF1, positive for the inhibitory marker PD-1, and this is crucial negative for a more terminal exhaustion marker called TIM-3.
That combination of markers tells a story, doesn't it? What does being TCF1 positive actually mean for a T cell?
TCF1 positivity is the key to their stem-like nature. It's a factor associated with memory, with self-renewal, with progenitor capacity. These TSL cells are the ones that can replenish the exhausted T-cell pool.
They express PD-1, so they're still being stressed by the chronic infection?
Right, the brake is still on. But because they are TIM3 negative, they haven't yet differentiated into those terminally exhausted, essentially useless T cells. They're exhausted, yes, but they're reinvigoratable.
They're the only ones capable of mounting a sustained, long-term anti-HIV response if we can just remove that immune brake.
That's it. And their regulation and function during HIV infection was, until now, not very clear, which makes this finding so important.
Okay, that clarifies the target. So let's dive into the results of the PDC depletion in these humanized mice. What was the first, most direct piece of evidence that this intervention was working on the inflammation?
The first and most immediate finding was the impact on those alarm signals. Depleting the PDCs significantly lowered the expression of the type I interferon genes, specifically IFNA2 and IFNB1, in the mouse spleen and other tissues.
So that immediately confirmed the PDC's role in sustaining that residual inflammation.
It did, but they went a step further and measured what are called interferon-stimulated genes, or ISGs, things like MX2 and ISG15. These genes are the downstream response, the cellular manifestation of the alarm going off.
Why measure those two genes specifically, MX2 and ISG15?
They're classic, highly reliable biomarkers of IFNI signaling. MX2 is a potent antiviral protein that's induced specifically by type I and type 3 interferons. ISG15 is another critical protein that modifies other proteins inside the cell, basically shifting the whole cell into an antiviral state.
So by seeing a significant drop in those, they confirmed that the functional effect of the chronic signaling was gone once the PDCs were removed.
The alarm noise was gone, exactly.
So did silencing the alarm then lead to the recovery of the T cells?
Yes, and this was one of the most exciting results. First, PDC depletion reversed those markers of immune hyperactivation we talked about earlier. The frequency of cells co-expressing CD38 and HLA-DR was reduced.
Meaning the T cells were no longer stuck in that state of chronic non-productive inflammation.
Right. And they also saw a reduction in unwanted T cell proliferation and, critically, a reduction in apoptosis cell death in the CD8 plus T cells. They were just surviving better in this now less toxic environment. And this led to a full restoration of both CD4 plus and CD8 plus T cell numbers compared to the control mice.
A true immune reconstitution.
It looked like it.
So the PDCs weren't just causing generalized inflammation, they were actively contributing to the death and hyperactivation of the T cells. How did the T cells look functionally after this? Did they regain their ability to fight?
They absolutely did. This is the functional rescue part. When the researchers stimulated the spleen cells from the PDC-depleted mice with HIV peptides, so they gave the T cells a target to recognize, they were able to mount a significantly better antiviral response.
What did that look like?
They saw increased production of key antiviral cytokines, specifically interleukin-2, or IL-2, and interferon gamma, or IFN gamma.
And why are those two in particular so important?
Well, IFN gamma is the classic killer molecule, essential for direct cytotoxic activity. But IL-2 is the growth factor that promotes T-cell proliferation and longevity. Seeing an increase in both suggests the T-cells weren't just ready to fight, but they were ready to sustain that fight.
And this rescue was specific to the HIV response.
That's a crucial point. When they stimulated the T-cells non-specifically, the improvement was much less pronounced. This confirmed that the PDCs were specifically suppressing the targeted anti-HIV response, not just causing generalized fatigue.
Which brings us to the ultimate goal, the real litmus test, reducing the viral reservoir. Did rescuing the immune system actually manage to shrink the amount of hidden virus?
Yes. And this is the finding that could fundamentally change therapeutic strategy. PDC depletion led to a reduction in the amount of cell-associated HIV DNA. That's the measure of the integrated viral remnants, by 3 to 5-fold.
And this was across multiple tissues?
Across multiple tissues. Lymph nodes, spleen, bone marrow, liver, lungs, and even more, the depletion reduced the number of cells that harbor replication-competent HIV, which they measured using the gold standard viral outgrowth assay.
A 3 to 5-fold reduction is significant in a research context, but I have to press you on this to keep our listeners grounded. The viral reservoir is massive. A 5-fold reduction is great, but it still leaves a huge amount of virus behind. How significant is this biologically in terms of actually achieving a functional cure?
That's a critical point of interpretation, and you're right to ask. In the world of HIV cure research, a 5-fold reduction from a single intervention like this is absolutely massive. Because most monotherapies, single drugs or agents designed to shock the virus out of latency, have historically failed to show any substantial reduction in the reservoir size in vivo. This study showed that simply by removing the inflammatory brake, the host's own immune system achieved this reduction.
So it proves two things.
It does. First, that the immune system, when it's rebalanced, is highly capable of tackling the reservoir on its own. And second, it suggests that this persistent inflammation isn't just an annoying side effect of HIV, it is the active biological mechanism that prevents the immune system from establishing control.
Which opens the door for combination strategies.
Which we'll get to. But this is a powerful distinction. Was this reduction just a side effect of removing the inflammation, or was it directly dependent on those rescued elite T cells actually doing the killing?
Right. Did the T cells do the work?
This is where the mechanistic elegance of the study really shines. It's arguably the strongest piece of evidence they have. They ran a definitive control experiment where they simultaneously depleted both the PDCs and the CD8 plus T cells.
Using a specific antibody, OKT8, to get rid of the CD8 plus T cells.
Correct. And what they found was that the reduction in the HIV reservoir did not happen if the CD8 plus T cells were missing, even though the PDCs were gone.
That is the ultimate mechanistic confirmation.
It is. It confirms unequivocally that PDCs were promoting HIV persistence by suppressing the CD8 plus T cells. And it was the rescue and subsequent activity of those T cells that was responsible for eliminating the reservoir cells.
The CD8 plus T cells are required.
They are absolutely required. The experiment didn't just show a correlation, it showed a mandatory mechanism. And they even went further and correlated the size of the reservoir with the recovery of those specific stem-like CD8 plus T cells.
The TSL cells.
The TSL cells. And they found a highly significant negative correlation. The more of these fully functional TCF1 positive elite fighters you had, the smaller the viral reservoir became.
Okay, so we've established that PDC depletion is effective on its own. It removes the immune brake, it restores function to these crucial TSL cells, and it shrinks the reservoir. Now let's move to the idea of synergy. If you've successfully woken up these highly potent T cells, you should be able to hit the gas too.
Which leads us directly into combination therapy. That is precisely the logical leap the researchers made. They recognized that the stem-like CD8 plus T cells they had rescued, the PD-1 positive cells, are the primary target for a powerful class of cancer therapies.
The immune checkpoint inhibitors, or ICIs.
Exactly. Specifically, they looked at PD-1 blockade using a drug like pembrolizumab, which is widely used in oncology.
So explain how these immune checkpoint inhibitors work. This is the hit the gas part of the equation, isn't it?
It is. In cancer and in chronic viral infections, T cells become exhausted and they express these inhibitory molecules on their surface, like PD-1. You can think of PD-1 as the molecule that tells the T cell, stop fighting, the risk is too high.
It's the molecular brake.
It's the molecular brake. When a corresponding molecule, PDL1, binds to PD-1, the T cell is shut down. By blocking PD-1 with an inhibitor like pembrolizumab, you remove that inhibitory signal. You release the brake and you reinvigorate the T cell, allowing it to respond aggressively again.
The rationale for the combination seems perfect then. PDC depletion clears the toxic inflammatory environment, which increases the number and the quality of the TSL cells. Then the PD-1 blockade takes those healthy but still inhibited TSL cells and just turbocharges their ability to kill.
Exactly. The combination is designed to maximize the potential of the host's own immune system. The two therapies target different but interconnected aspects of T-cell dysfunction. One addresses the environmental cause, the inflammation, and the other addresses the molecular consequence, the inhibition.
So what did they find when they tested this combination in vitro, using blood samples from people living with HIV who were already on stable CART?
The in vitro results really confirm the synergy. They saw that PDC depletion alone and PD-1 blockade alone both individually increased the frequency and number of what are called polyfunctional CD8 plus T cells.
And polyfunctional just means they're the strongest fighters, right? They can do multiple things at once.
Right. They can produce multiple antiviral molecules like IFN-gamma, TNF-alpha, and IL-2 simultaneously. But the real magic happened with the combination. PDC depletion plus pembrolizumab led to a cooperative enhancement. It further increased the frequency and number of these highly effective polyfunctional stem-like T cells.
Does it show they work better together?
Much better together.
And did this strong synergy translate? when they moved the combination back into the humanized mouse models in vivo.
It absolutely did. In the humice, the combination treatment provided a superior enhancement of the anti-HIV T-cell responses. They saw a significantly greater increase in T-cells producing IFN gamma and TNF-alpha compared to either treatment alone.
The TSL cells were fully rescued and aggressively activated.
They were. And most importantly, when they looked at the viral reservoir, the synergy continued. While PDC depletion alone was effective, reducing the reservoir by that 3- to 5-fold, adding pembrolizumab led to a further reduction of cell-associated HIV DNA.
And was this reduction seen everywhere?
It was particularly notable in the bone marrow tissue, which is often a really tough-to-reach reservoir site.
So we have a clear step-by-step model here. Step 1, PDC depletion clears the chronic inflammation, reducing T-cell death. Step 2, this rescues the elite TSL cells. And step 3, PD-1 blockade then turbocharges those specific rescued cells, leading to a synergistic, deeper attack on the reservoir. This feels like a complete conceptual shift.
It really is. This research suggests a completely new target for HIV curative strategies. For years, the focus of the cure effort was on the shock and kill strategy.
Using latency reversal agents to force the dormant virus to wake up, hoping the immune system could then kill it.
Right. And that strategy has been hampered because the T cells are just too exhausted to kill the awakened virus effectively.
But this new approach focuses on immune rebalancing first.
It says, fix the immune system first. Remove the chronic brake, the PDCs, and only then activate the T cells with PD-1 blockade. This prepares the host's immune system to control or possibly eliminate the persistent virus, regardless of a shock phase.
That shift in focus from purely attacking the virus to repairing the environment is one of the most significant implications here. They're showing that treating the chronic inflammation is a viable way to shrink the reservoir.
Because that inflammation is what's impairing the T cells you need for control.
And this PDC-driven mechanism, this type I interferon axis, it has implications far beyond HIV. You mentioned other inflammatory diseases. Where else is this mechanism at play?
The broader implications are vast. The sources confirm this type I interferon mechanism driven by PDCs is implicated in several other chronic inflammatory and autoimmune diseases. The most notable one is systemic lupus erythematosus, or SLE,
Lupus.
and related cutaneous lupus conditions. In these diseases, the overproduction of IFNI by PDCs is a central driver of the pathology.
That's fascinating. Does that mean that therapies targeting PDCs are already being tested in humans for these other conditions?
They absolutely are. And this provides crucial translational context for these HIV findings. Because the BDCA2 marker is specific to PDCs, pharmaceutical companies have already developed and tested BDCA2 targeting antibodies for treating SLE.
So there's a precedent.
A huge precedent. Clinical trials targeting PDCs have already shown efficacy and, critically, an acceptable safety profile in human patients. In trials for lupus, BDCA2 inhibition has been reported to significantly reduce the interferon gene signature and improve disease symptoms with an acceptable safety profile over 50 weeks or more.
So we already have early data suggesting that selectively removing or inhibiting PDCs in humans is feasible and tolerable.
Which bypasses a major hurdle in translating novel HIV findings from the lab to the clinic.
And there's also a strong connection to cancer, which uses similar immune evasion tactics to survive.
Indeed. The connection lies in that T-cell exhaustion pathway. Tumor-associated PDCs have been found in various human cancers, and they're frequently associated with a worse prognosis for the patient. They infiltrate the tumor microenvironment and contribute to the immune suppression that allows cancer to thrive.
So the findings from this HIV study suggest this strategy could be a valuable addition to existing cancer immunotherapies.
Exactly. In oncology, the goal is often to flood the system with activated T cells using PD-1 blockade. If PDCs are acting as a local inflammatory brake in tumors, eliminating or reprogramming them could significantly enhance the efficacy of existing PD-1 inhibitors for cancer patients, regardless of their HIV status. You're tackling a common, fundamental inflammatory brake in chronic disease.
So this research presents an incredibly promising pathway forward, essentially borrowing techniques proven in oncology and rheumatology and applying them to virology. But as we always do, we need to ground these findings in reality and discuss the limitations. What are the key caveats here?
Well, the primary limitation, which the researchers acknowledge up front, involves the model itself. Most of the detailed in vivo work was done using those humanized mice.
The HU-BLT models.
Right. And while these models are invaluable, they support HIV replication, persistence, and responses to CART in a way that's similar to humans, they still have known structural and functional pitfalls.
Can you elaborate on the most significant pitfalls, the ones that might impact how we interpret this reservoir reduction data?
Certainly. These humides have, for example, disorganized lymphoid tissue structures. This might affect how T cells and PDCs interact spatially, which could influence the inflammatory signal in a way that doesn't perfectly mirror a human lymph node.
And there are other impairments too.
Crucially, they also have impaired B-cell maturation and suboptimal human myeloid cell function. So while the core immune mechanisms involving T cells and PDCs appear accurate, the overall complexity of the human immune environment isn't fully replicated. We have to be cautious when quantifying viral reduction in a mouse model.
And then there's the major leap, the translational uncertainty from the lab to the patient.
Exactly. The findings that were confirmed using blood samples, the PBMCs, they lack the complex, continuous immune microenvironment of lymphoid tissues that you see in vivo. An in vitro assay just doesn't perfectly capture the ongoing viral dynamics or the complete cytokine environment present in a real human lymph node or bone marrow.
Which are the primary reservoir sites?
They are. So the safety and feasibility of PDC depletion, especially in combination with PD-1 blockade, needs rigorous, careful consideration before clinical trials can begin in people living with HIV.
Finally, there's a technical trade-off here. This study chose to target the cell producing the interferon, the PDC, but other researchers looked at just blocking the receptor for interferon. What are the pros and cons of those two approaches?
That's a crucial distinction about specificity. Previous research has shown that blocking the type I interferon receptor, IFNAR1, can also rescue T cells and reduce the HIV reservoir.
So a similar result.
A similar result, but the trade-off is the loss of generalized immunity. Blocking IFNAR would stop the activity of all type I interferon produced from all cell types, and those are necessary for fighting other pathogens like the flu. Blocking the receptor wholesale might impair overall antiviral immunity during the treatment.
So targeting the PDC is a more surgical and potentially safer way to manage the chronic inflammation without destroying acute antiviral defenses.
Precisely. The target they used, BDCA2, is expressed specifically on human PDCs, which are a small, defined population. This means the intervention is highly specific to the cell type that's driving the chronic inflammation. Depleting them temporarily is far less immunologically disruptive than completely silencing the entire IFNI signaling pathway across all cell types. The specificity is what makes it so exciting for clinical translation.
So we've taken a comprehensive deep dive into the dual nature of one of the immune system's most critical, yet sometimes detrimental players, the plasmacytoid dendritic cell. We've learned that chronic inflammation sustained by these overactive PDCs constantly sounding the type I interferon alarm is the fundamental mechanism weakening those elite fighter T cells and protecting the hidden HIV reservoir.
And targeting these inflammatory PDCs successfully rescued the crucial stem-like CD8 plus T cells, the TCF1 positive warriors, which led to a measurable and significant reduction in the reservoir.
And then combining this PDC depletion, which is like releasing the immune brake, with PD-1 blockade, which hits the gas, offered a synergistic and enhanced way to shrink that viral reservoir even further.
Right. As Dr. Su noted in the paper, the immune system's continued effort to fight the infection can sometimes work against itself. And this research is a powerful demonstration that immune conditioning, specifically by surgically clearing out the chronic inflammatory cells, can restore the host's ability to control or potentially eliminate the persistent virus.
It moves us past that old paradigm of simply killing the virus
and into the new era of fixing the immune environment itself.
This study really does fundamentally shift the focus of HIV cure research. It treats the persistence of the reservoir as primarily a problem of localized chronic immune inflammation and T-cell exhaustion, rather than an insurmountable viral challenge.
Which leads us to a provocative thought, one that extends far beyond just HIV.
If the key to treating the long-term persistence of HIV is safely and temporarily adjusting these specific immune cells to interrupt chronic inflammation, what other chronic diseases associated with persistent low-grade inflammation, like certain autoimmune conditions or difficult-to-treat cancers, might yield to this same strategy?
The strategy of immune rebalancing.
Exactly. Future studies, building on the safety data from those SLE trials, will determine if this can safely achieve a functional cure for HIV. But the underlying principle suggests a powerful new tool in the immunological arsenal for treating chronic disease everywhere.
Thanks for tuning in to the Health Headliner podcast by To-Do Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.